Hi, everyone. My name is Chinchin Yu, and I'm a fifth-year grad student in the Halachek Lab, and I'm honored to be introducing our colloquium speaker today, Dr. Nikta Fakhri. Dr. Fakhri is an associate professor in the Department of Physics at MIT and the Physics of Living Systems group. Her lab's research spans physics and biology with a focus on studying the non-equilibrium dynamics of living systems. Dr. Fakhri completed her undergraduate degree at the Sharif University of Technology in Tehran, Iran. She then went on to get her PhD from Rice University, and afterwards she was a Human Frontier Science Program postdoctoral fellow at the University of Göttingen in Germany. She joined the faculty of MIT Physics in 2015 and was promoted to associate professor in 2020. Congratulations, Dr. Fakhri. Dr. Fakhri has won numerous awards, including in 2017, an Alfred P. Sloan Research Fellowship in Physics, in 2018, the IUPAP Young Scientist Prize in Biological Physics, and in 2019, the NSF Career Award. Dr. Fakhri has worked on numerous systems, ranging from the mechanical dynamics of nanotubes to pattern formation in starfish oocytes. And for me, one of the most exciting parts of her research is that she is able to apply principles from non-equilibrium physics to test models in oftentimes very messy biological systems. We are very excited to hear about her group's latest work today, and please join me in welcoming Dr. Fakhri.
Thank you, Chinchin. I will share my screen now. Okay. Thank you, Chinchin, for the introduction. It's a great pleasure to be hosted by the Society of Women in the Physical Sciences for my virtual visit. And I wish it was in person, but so far I had a wonderful time meeting with both the students and faculty. And today I'm going to talk about my group's research and our fascination with symmetries and symmetry breaking processes in non-equilibrium living systems. And I, like many other physicists, am fascinated by the fact that there really is something different about being alive. And we'd like to know what this is, in the same way that we know what it is for a collection of atoms to be solid and for a collection of electrons to be superconducting. Of course, the what is life question goes back to Schrödinger, and one might think that the molecular biology would have answered this question by now. When we look around, more or less immediately, we can identify things that are alive, and usually the criteria that we use in making this discrimination between animate and inanimate matter have nothing to do with DNA or proteins. Being alive is a macroscopic state, while things like DNA and proteins are components of the microscopic mechanisms by which this state is generated and maintained. So let's talk about this video that you see on my slide. This is an egg cell or oocyte of a starfish, a model system that we use in my group, and I'll showcase that throughout the talk. What you see is self-organization of proteins that allow the cell to perform a very important function at this very first stage of development, namely cell division. And as you can see, the patterns continuously self-organize from one state to another. And this is a quote that I really like from Alan Turing's paper on morphogenesis, which says most of an organism, most of the time, is developing from one pattern into another. And I think this really highlights the importance of self-organizing principles rather than molecules. Of course, molecular biology have been very successful in identifying the molecular players and the microscopic mechanisms, but I think we have made little progress in identifying the order parameters that are characteristic of the macroscopic state. So I think for me, the question of what is life is really the question of what are the order parameters that are characteristic of the living state. And as we know in equilibrium physics, the concept of order parameters and symmetry breaking has been very powerful, and they have played a crucial role in defining a wide range of phenomena. For instance, symmetries at the microscopic scale govern the bulk response of materials, and they fundamentally define the constitutive relations that describe the way in which, for instance, waves propagate in materials. Now, asking for the order parameters of living state is obviously a hard problem, and I admit that it's not a very well-posed question. However, I think it is crucial if we would like to imagine a physics of living systems that reaches the level of predictive power that has become the standard in many areas of physics. And today, I like to highlight my group's approach in identifying symmetries and symmetry breaking processes. And I will talk about the experimental tools that we developed to observe non-equilibrium degrees of freedom, the data analysis frameworks to define order parameter fields. And then finally, I'll highlight our collaborations with my theory colleagues to develop theoretical frameworks which have predictive power. And to showcase our approach, I will highlight 3 examples of broken symmetries that we have quantified. First, I will talk about the most important symmetry, in my opinion, that is the defining feature of non-equilibrium systems, namely time reversal. And what I will show you is how we can use tools from stochastic thermodynamics to quantify the thermodynamic arrow of time and dissipation. I will then move up the scale as well as complexity, and will show you how biochemical signaling proteins, like the ones that I show you in the videos, self-organize at cellular scales, and how we can use concepts from topology to uncover these self-organizing principles. And then finally, I'll talk about something that I'm very excited about, and show you how time reversal asymmetry and non-reciprocal interactions can give rise to collective dynamics at organismal and population level. And for each of these examples, I will highlight the experimental tools that we developed, the data analysis algorithms, and also the theories that will allow us to get a better understanding of these principles. Okay, so let me start by what is arguably the most important symmetry breaking in non-equilibrium systems, time reversal. And to do that, let me start by showing you a video of a developing embryo of a starfish. In the first image here, you can see many cells. And to orient ourselves, the cell nuclei is marked in blue. So I'm going to start the video and play it for a bit. And I'm sure the majority is beginning to realize that there is something unusual about this video. And sooner or later, you will recognize the fact that I'm actually playing this video backwards. And I think this highlights a fundamental feature of living systems, which is at the expense of dissipated energy, living systems perform irreversible processes that allow them to propagate and reproduce. And as dictated by the second law of thermodynamics and its fluctuation theorem, irreversibility in non-equilibrium processes can be quantified in terms of how much entropy such dynamics produce. And a recent beautiful theoretical result from Juan Parando and others have shown that there is an exact relationship between the entropy production and the distinguishability of a process from its time reverse, quantified by relative entropy between the forward and backward states. And given the fact that this is actually my favorite equation at the moment, let me take a little bit of time to go over the details. So what you see here is the dissipated work that has been written in terms of a well-known and powerful concept from information theory, the relative entropy or the Kolbeck-Leibler distance between 2 probability densities. And when we write it in this way, this result actually reveals a very deep meaning, which links the dissipation directly to the irreversibility of the underlying experiment. And generally speaking, the relative entropy between probability densities is expressing the difficulty of distinguishing sampling from 2 densities. And in this present form, it actually measures the difficulty of distinguishing whether observed data corresponds to those from a forward or backward experiment. So this relative entropy can be considered as a quantitative measure of the arrow of time. And again, this relationship is actually very general. It's shown to be valid for a wide family of initial conditions. It's also valid for both classical and quantum systems. Now, I want to highlight something important, and that is the fact that often in experiments, only one or several of the non-equilibrium variables can be tracked in time, which means that if we apply this to experimental data, this equation only yields a lower bound for the entropy production rate. And I'm going to call that lower bound small sigma, or irreversibility. Now, of course, the video of the developing embryo that I showed you is an unmistakable example of an irreversible process, namely cell division. But time reversal symmetry breaking happens at much smaller scales, and it manifests that system dynamics at much larger scale. So let's look at intracellular scales, where non-equilibrium activity is mostly dominated by stochastic force fluctuations, and in particular, a structure known as actomycin network. And then before I continue to tell you how we quantify irreversibility, let me just give you a quick refresher on the actomycin network in case you are not familiar with it. So the actomycin cortex is a quasi two-dimensional structure, dissipative structure of dynamically cross-linked actin filaments that continuously polymerize and depolymerize. And in this video, for instance, you can see actomycin cortex again of the starfish oocyte, and actin is labeled in red. And this network is anchored to the cell membrane, and it's internally activated by myosin motor proteins. So you can think of myosin motors as force dipoles that generate stochastic force fluctuations in this structure. And the cortex is actually quite an important structure in the cell because it provides mechanical integrity and rigidity to cells over extended timescales. However, its component turnover on the order of minutes. And in addition, the cortex undergoes reorganization during cellular processes such as cell division, which you see an example of that in this video, but also generation of polarity and motility. And let me just kind of highlight a few important points. One is the fact that in this system, energy injection happens continuously at the molecular scale, which means that these systems are a class of active matter. And this energy injection gives rise to the spontaneous self-organization of the structures, which are examples of non-equilibrium steady states. And I want to point out that this is fundamentally different from non-equilibrium processes in other physical systems, where we drive the system away from equilibrium and look back at its relaxation to equilibrium. This is actually a true non-equilibrium steady state. And which means that these systems you can basically observe emergence of longer spatiotemporal scales. For instance, again, as I point out, the mechanical integrity of the cells is on the order of hours. So the cell has to keep its structure intact over hours, but the molecular turnover rate is on the order of minutes. And processes such as cell division, as you can see, happens on the scales of tens of microns, whereas the molecular scales is on the order of tens of nanometers. So now, I guess the question to ask is, how do we measure fluctuations of systems like this, and to be able to quantify this time reversal asymmetry? So to do that, to basically be able to quantify time reversal asymmetry, we need long time series data of intracellular force fluctuations with high spatiotemporal resolutions. Because again, I want to point out that these are probability distributions, so you really need a lot of data to construct these distributions. So my group has been developing various tools to measure fluctuations, and I'm going to give you one example today. So in the past, we've been using single-walled carbon nanotubes as probes, but recently we have discovered, actually very serendipitously, fluorescent granules in the cortex of starfish oocytes that emit above 900 nanometer in infrared. And they're about a micron in diameter, and they're highly photostable. So you can image them for very long periods of time. And these probe particles are embedded in the cortex. And this is an example of a video, as you can see. So we have many of them in the cortex, and the irreversible noise that acts on them originates basically in the jostling of these myosin motors, which are force dipoles. And so what we can do is we can collect large position time series data of many, many particles to be able to quantify irreversibility. So to estimate irreversibility, we need data that's obtained from stationary steady state process. And in this system, the underlying dynamics of the actomycin network is stationary, which leads to stationary local force fluctuations acting on our proparticle. And considering that the dynamics of the proparticles are overdamped, we can interpret changes in position as readouts of these stationary local force fluctuations of the underlying non-equilibrium atomizing network. And we calculate these position increments using an adjustable lag time, which are called theta. And this lag time is actually a very important part of our analysis, as it becomes clear in my talk later. But just to give you an intuition, larger theta corresponds to more coarse-grained data. So basically, by varying this theta, we are generating a collection of observables whose estimated KL divergence captures irreversibility on scales larger than theta, the scales that have not been smeared out by coarse graining. Okay, so before I tell you how we kind of construct this probability distributions and KL divergence, let me give you an intuition of what we should expect from the dynamics of the system. This is a system that we understand quite well. So it basically tells us, gives us the ability to really interpret the irreversibility measure. So to do that, we are going to look at the normalized time lag velocity autocorrelation function. And as you can see, the velocities are delta correlated at time t0, and then they're negatively correlated for small delta t's with magnitudes that diminish for longer lag time. And this is consistent with probes dynamic being driven primarily by thermal fluctuations in a locally elastic network on short time scales, which means that probe particles experience a negative velocity that returns them to their initial position. But on longer time scales, our non-equilibrium activity allows the particle to basically diffuse away. And this is actually consistent with the previous work that I and others have done, where we show Meisen force fluctuations result in active steering of cytoskeleton as timescales larger than around 1 second, peaking at about 10 seconds, which is Meisen's on and off timescale. And what you can see in this mean square displacement figure as a function of lag time is basically at timescales of more than 1 second, you have this violation of fluctuation dissipation theorem. So your fluctuations are considerably higher from what one would calculate from the response function if FDT holds. So now I'm going to corroborate this conclusion by demonstrating the emergence of this timescale when we analyze our multi-scale irreversibility, which actually offers us a model-independent verification of these timescales that are related to the non-equilibrium dissipation or entropy reduction in our system. And without kind of going into details, I'm happy to talk about this later during Q&A. We developed an algorithm to measure KLD between the forward and reverse velocity time series data. And we did that for each coarse-grained time series. And the main result is basically an estimate of the irreversibility as a function of lag time theta. And this is a plot that I absolutely love, because what you can see is that the estimated irreversibility is a non-monotonic function of the lag time with a peak between about 1 to 10 seconds. And the way we understand this non-monotonic behavior is basically as follows. So for large time scales, basically we have coarse-grained out too much information, which generically leads to smaller KLD and therefore irreversibility estimates. For short time scales, the estimated irreversibility is small, which basically the reversibility at short time scales is consistent with velocity statistics being dominated by the high frequency fluctuations coming from the reversible thermal noise. So we're really probing thermal fluctuations. But then the irreversibility noise acting on our probes originates in the contracting and jostling of these Meisen motors, whose effects on the probe requires longer time to manifest. So what this non-monotonic behavior of KLD is telling us is that as we probe progressively long time scales by increasing the time lag, we first capture reversible thermal noise, followed by an increase at longer scales due to emergence of the irreversible active noise. And eventually we're coarse graining away too much information, resulting in smaller observed irreversibility. So this peak really represents a trade-off between timescales at which irreversibility is most prominent and the suppression due to coarse graining. And I find this absolutely exciting because we infer this timescale using a model-independent irreversibility metric, which allows us to characterize the nature of the dissipation in the system. Now, if you kind of notice and remember what I mentioned before, is irreversibility is a lower bound number. And the question is, how good is it? In other words, if I prepare the system in different conditions, can we distinguish different activity levels with this lower bound number? And to kind of demonstrate that, we're going to look past the dissipation timescale, where we know that our estimator is capturing the detectable irreversibility of the process. And this observation allows us to infer relative irreversibility of systems with different amount of non-equilibrium activity, which we can actually, it's a biological system, which we can achieve either biochemically by overexpressing myosin, our forced dipole, or by depleting ATP to generate a low activity steady state. And as you can see, we've been able to verify this, where here we can see that I plot the estimated irreversibility as a function of lag time for these 3 treatments of oocytes, high, medium, and low activity, and each for multiple cells. And despite the fact that these estimates all decrease with lag time, they clearly appear clustered into 3 zones corresponding to the 3 activity level. And despite the fact that KLD is only offering a bound to physical dissipation, our probe particles are maintaining this monotonic relation between the detectable irreversibility and the expected non-equilibrium activity. And again, this is really exciting because now we can try to really understand the underlying mechanisms of the dissipation in the system. Okay, so now that we see there is this notion of dissipation scale in living systems, the question is, how does the cell tune these spatiotemporal scales? How does these longer spatiotemporal scales emerge? And just like we turn on external fields in physics to change correlation lengths, for instance, in an Ising model, cells use biochemical fields to tune spatiotemporal length scales. And here I'm going to show you 3 videos of our probe particles in the actomizing cortex. And in the first video, you can see that the dynamics of probe particles are highly stochastic. So you just see kind of what appears to be Brownian motion. And in the second video, you can see traveling waves of the particles. And then finally, in the last video, you see spatiotemporal chaotic dynamics of these probe particles. And what's happening is basically by patterning the biochemical field, you can markedly get different dynamics of actomizing networks, which then correlates with the particular function the cell needs to perform. And the master regulator of actomyosin network is a signaling protein known as Rho-GTP. And each of these proparticle patterns of movement that I'm showing here corresponds to a particular Rho-GTP biochemical pattern. And this brings me to the second part of my talk, which is basically I'm going to move up the scale as well as complexity. And I'm going to show you how biochemical signaling proteins self-organize at cellular scale and how we can use concepts from topology to understand these self-organizing principles. And just to kind of like give you a quick refresher, we know that chemical waves constitute a very well-known class of dissipative structures, which normally emerge in reaction diffusion systems. And in a biological system, they actually play a very important role because they spread information rapidly to synchronize and coordinate biological events. And here you see the video of a starfish egg cell, again, my favorite model system. It's went too far. And what you can actually see is during meiosis, which is the process by which cell undergoes asymmetric cell division, the cell shape is dynamically deformed by surface contraction waves that travel along the membrane from one pole of the cell, which I mark here as a VP, to the other pole, which is marked by AP. And surface contraction waves are induced by the signaling protein Rho-GTP, which is when GTP bound locally triggers atomizing contractility, and thus it generates this zone of surface contraction wave that travels as a band across the membrane. And recently, in a new work that's coming soon from my group, we have shown the key mechanism for this Rho-GTP pattern formation. So just to give you a very quick intuition, Rho is a protein with two states. When it's in inactive form or GDP bound, Rho can either be a membrane bound or cytosolic. And when it binds to the membrane, it undergoes nucleotide exchange, which converts it into an active GTP bound state. And this GDP to GTP conversion is mediated by activator proteins known as GEF. For starfish, it's EG2. So I'm just throwing out this name because I'll be using it later in the talk. And when GTP is hydrolyzed by another protein known as GAP, it results in its detachment from the membrane. And what we were able to show was that GEF-EG2 is a bistable switch, and its front demarcates subdomain on the membrane with high and low nucleotide exchange rate. So now if you imagine we are initially in a low EG2 concentration, basically, which is the point A1 on this phase portrait, such that most of the Rho is in inactive or GDP bound form. So if you go through a sudden increase in the EG2 concentration, this shifts the steady state towards an increased Rho-GTP and decreased Rho-GDP concentration of the membrane, which is 0.3 on the face portrait. But because we have positive feedback on Rho activation, the Rho concentration do not relax directly into its new steady state, but it actually transitions via a large excursion in the phase space, going from 0.1 to 2 and 3. And basically what we were able to show was that Rho dynamics is excitable. Now, biochemically, it's possible to increase the amount of EG2, our activator in the cell, which we, for instance, do it via overexpression. Now, instead of now the local Rho kinetics is no longer excitable, but it exhibits limit cycle oscillations because excitability and bistability are actually very close in the phase space. And this now results in local oscillations of the Rho-GTP concentration on the membrane confined to the subdomain with high EG2 concentration. And what this means is that we now have a formation of a range of different patterns, not just traveling waves, and in particular, these beautiful spirals that you can see in this video here. But this is actually quite exciting because what it means is that we have a biochemical knob to basically tune the Rho-GTP dynamics in the cell and generate this dictionary of different patterns. And here I'm showing you 6 of them here. The fluorescence intensity that you see is proportional to the concentration of the Rho-GTP on the membrane. And each of these steady states basically represents a different spatiotemporal wave pattern. And as you can see, the typical wavelengths range from 15 to 30 micron. And for the rest of this talk, I'm going to refer to these steady states at state 1 to 6. And I guess one thing you just need to remember from this slide is as we go from state 1 to state 6, cellular activity is increasing, and then the states appear to be more chaotic. Okay. So as I mentioned, to understand kind of like this self-organizing principles in living systems, my group searches for order parameters. And in this system, the way we define order parameter is basically as follows. So as a Rho-GTP wave pass through a point on the 2D membrane, so imagine you are a point on this 2D membrane, they produce an oscillatory fluorescent signal. And from the time series of the pixel intensity oscillation, we can extract the relative phases across the membrane. And then what happens is that now with this analysis, we actually have a phase field view associated with the Rho-GTP concentration field. So now you can think of this whole kind of oocyte as being a collection of oscillators that are diffusively coupled. And these phase fields, the first thing to notice is they harbor a dense population of topological defects. Topological defects are distinctive singularities that are fingerprints of the broken symmetry field. The defects can be analyzed by looking at the local wave vector field, which is the gradient of the phase field. And the charge of the defect is given by an integer valued winding number. And the phase fields we have in this experimental data is generally have winding number of +1 or -1, or +1 and -1 are the most stable ones. And these correspond to clockwise or a counterclockwise rotating local phase field dynamic. And what you see is localized creation and annihilation events always involves pairs of oppositely charged defects. And this actually results in the conservation of the total topological charge. So now the dynamics of this really complex biochemical field can be analyzed in terms of a gas of +1 and -1 charged particles. Okay, so let me give you an intuition of what this order parameter field is. So we can understand the phase velocity basically as an effective velocity field that describes a potential flow with vorticity omega localized at this point-like vortex cores. And one thing to remember is that these are signaling proteins. So this local wave vector field is proportional to the velocity at which the information encoded in the concentration waves are propagating on the 2D membrane. So we're really looking at how information is getting transmitted across the cell. And one kind of interesting observation is the phase field defects of this charge +1 or -1 actually maps to the centers of this counterclockwise or clockwise vortices in the phase velocity field. And what we observed was the fact that the system exhibits turbulent-like behavior, which is characterized by a weakly correlated distribution of a large number of vortices and anti-vortices of unit winding. And if you look at the system, we can actually identify dipoles that are persistent, but also dipoles that are transient. And if we kind of quantify the relative fraction of persistent dipole pairs for each of the steady states that I showed you before, what you can see is that they all are characterized by a low fraction of bound vortex anti-vortex pair, about 20%, which is actually reminiscent of the disordered phase in a BKT topological phase transition. And when we looked at different scaling laws also for the system, for instance, velocities, and I'm not going to go into details of that, but all of these statistics are consistent with having topological turbulence in this biochemical pattern, signaling patterns. Okay, so while the phase defects or phase velocity vortices corresponds to topological structures in this two-dimensional cell membrane, there's actually another dimension, which is time. And to examine these structures in 2+1 space-time dimension, I'm going to switch to a view of the 3D phase field continuum, and I'm going to play this video. So now what you can see is the defect trajectories are extended into worldlines, and the pair creation and annihilation of the defects with opposite charges actually corresponds to positions where worldlines are joining and resulting in formation of loops. Also, if you look at the X time or Y time slice, you can see these beautiful structures, which actually I like to call time wrinkles. So as I mentioned, this creation and annihilation events are giving rise to formation of loops. And each of the signaling patterns that I showed you from state 1 to 6, they display a large number of closed loops. And some loops are simple loops, so they're created from single defect pair creation and annihilation, but some are actually very complex, and they can form by a large number of defect pairs exchanging partners. And as you can see, like an example of this here. But the striking observation is that the Rho-GTP spiral waves from all of these states actually exhibit varying frequencies of defect creation and annihilation, and hence loop densities, which correlates with cellular activity. So cellular, as you can see, as we go to higher cellular activity, we get more of this creation and annihilation event, and thus more of this loop structures. And obviously, this is the structure of the loops that we observe. But in addition to these short-lived loops, we also have, which mainly dominates when we have high activity states in our system, we also have low activity states, which instead of observing loops, we actually observe a large number of long-lived defect worldlines that exhibit spontaneous braiding dynamics. So space-time braiding of spiral cores is basically an indication of having chaotic dynamics in the Rho-GTP biochemical signaling pattern. But without going into kind of details and technical details, we can use a matrix representation of the braiding operator that basically denotes clockwise and counterclockwise crossing between neighboring particles. And then we can construct a braiding matrix. And then looking at the eigenvalues of this matrix, we can actually quantify the growth rate of topological entropy in a system like this. And what we see is basically topological entropy increases faster at higher cellular activity. And of course, what this results presents is basically a direct link between the spontaneous topological braiding of these spiral defects, but also the information loss in the cellular signaling waves. And again, as I mentioned, these signaling proteins, they contain information and they propagate information across the surface of the cell. Okay, so while you can see that this quantification of the global defect braiding basically suggests that we have a positive topological entropy growth rate for all of these signaling states, or in other words, basically we have information scrambling on kind of this much large scale. If we actually look at the nearby same sign defect pairs, what we can see is that pairs that basically braid deterministically on very small scales, and these basically braidings preserve information. So these detected pairs are consistently same sign charge, and they rotate around each other with a chirality that's actually consistent with the sign of their topological charge. And as they braid for 180 degrees and effectively change in the position, the phase field always return to almost an exact mirror of the prior rotation phases. And kind of an analogy to quantum mechanics, in the case when we have two identical quantum particles, the global phase shift that is basically acquired when we look at a counterclockwise 180 braiding operation determines if we have a bosonic, fermionic, or anionic particle for this fundamental excitations. But here, when we subtract this initial and final phase field after a 180 braiding event, we find that experimentally measured phase fields in this system almost always return to their initial configuration, which is actually consistent with having bosonic exchange symmetry. So, I mean, this is actually, in my mind, this is like very exciting because it reveals an unexpected parallel between our topological defects that are trapped in this two-dimensional membrane and a class of two-dimensional quantum particles that could be braided to encode and decode information. What's exciting is that in biology, we actually have a handful of tools to perturb and control this process. For example, we can deliver photosensitive domains into the biochemical circuit, which in principle we can use to braid the defects optically and perform computation using this cell membrane defects. Okay, so far, I guess I've been analyzing the system from a dynamical point of view. But since I brought up this idea of exploiting these self-organized structures of active matter for biological computing, I'm going to go back to the ideas from the first part of my talk and ask whether it is possible to use stochastic thermodynamics to quantify the work that's needed to create and maintain patterns like this, and what is the efficiency with which information exchange through these patterns happen. And actually, this is very, very exciting time for this stochastic thermodynamics and active matter, because a very recent result from Massimiliano Esposito's group have shown that basically there is a thermodynamic theory of reaction diffusion systems far from equilibrium. And basically they use stochastic thermodynamic and its systematic way to define quantities that we are used to, such as work and entropy, and they anchor them to the deterministic dynamics of the reaction diffusion systems. And similar to the Kolbeck-Leibler divergence for probabilities that I talked about in the first part of my talk, they actually introduce an equation that quantifies the dissimilarity between two concentrations. And what it demonstrates is that diffusive dissipation and mixing entropy can be fully described in a purely information theoretic fashion. And I think this theory will allow us to compute the work that's needed to manipulate a pattern and the efficiency with which information exchange happens through these traveling waves. And again, just going beyond a description, a dynamical description of the system, we can start thinking about, can we think about this phase transitions in terms of irreversibility, and using this irreversibility metric as an order parameter to look at these type of transitions. Okay, so also going to address another question, which is obviously whether, these ideas such as topology and stochastic thermodynamics allows us to understand from a physical point of view what these systems are doing, but do they allow us to drive a biological understanding of the system, and allows us to basically, would they allow us to pinpoint biological mechanisms? And let me first play this first video that I have, which is basically the biochemical field that I show you. And what we have done in my group recently is basically building tools that allows us to simultaneously image the biochemical field and the resulting, and basically the near infrared fluorescent probe particles. So we can look at the co-evolution of these two fields and the dynamics of the probe particles and presence of this biochemical fields. And we've been developing algorithms to extract strain fields from the dynamics of these probe particles. And now what we see is basically this multi-field view of the process. And there is this coupling between topological defects in the two fields. And I just want to remind you that these biochemical and strain fields are happening on a deformable manifold. And the cell uses the deformations that result from this mechanochemical coupling to change its shape, to divide, and to undergo morphological changes. So we can really think of these topological defects as organizers of biological activity. And I think this framework allows us to look at mechanochemical feedback mechanisms in this really complicated, messy biological systems. Okay, so in the last kind of part of my talk, I'm going to move even further up in scale. As you can see from the scale bars here, so now I'm going to talk about scales on the order of millimeters and hours, and talk about something that I'm quite excited about. And basically what I show you is how time reversal asymmetry can kind of propagate over much longer time scales in the system and give rise to collective dynamics. And I'm going to show you first a video. And before I play the video, let me just tell you what you are looking at. Each particle that you see here is basically an embryo of the starfish. So this is 24 hours post-fertilization. So the embryo has millions of cells. So the oocyte has undergone cell division. So now you have millions of cells. The cells on the outside, they have cilia, which is this organ that allows the cell to swim. The embryo breaks symmetry and starts swimming. And then when they come to the surface, they stand upright. And when you look at them from the top, basically there are spinner particles with a chiral clockwise rotation. Okay, so now I'm gonna play the video and you can see what happens. They say that a picture is worth 1,000 words, and this is a video, so you can do the math. But what you can actually see is this, they form this beautiful two-dimensional living crystal that actually undergo hexatic phase transition. And as you can see, as we play the video, which means that as we go through a development, they melt as a function of this developmental time. So this is a very new result from my group. Let me just give you an intuition of what we think is happening. So on the top, what you see is the shape of the embryos from the side and the corresponding flow fields. And what you see is basically individual embryos come to the water-air interface, and they enter a bound state at the air-water. And when we have sufficiently high embryo densities, the near-field interactions between rotating embryos lead to formation of a stable two-dimensional crystal with this hexatic order. And we can measure the flow fields around the embryos, and we now have evidence both from experiments and a theoretical collaboration with my colleague Jorn Dunkel in the math department that shows that you have Stokes slits that generate a hydrodynamic attraction between surface-bound embryos. And now what happens is, as the embryo develops further, the shapes become more elongated and the flow field changes. And we think that this actually results to this phase transition and the final melting of our chiral crystal. Okay, so let me show you some preliminary results on quantifying this order-disorder transition. Of course, we're fascinated by identifying order parameter fields. So you can define, for instance, an orientational order parameter that basically measures the degree of six-fold bond ordering in this hexatic phase. And what you see is basically a top row that shows the magnitude of this six-fold symmetry order parameter. And the bottom row basically shows the phase. And in the middle, what you see is a chymograph of the angle distribution as a function of time. And you can see that we can identify these stages of order to disorder transition. Again, these are very preliminary results, but this is basically a way of looking at how order to disorder transition happens in a system like this. But what's actually I'm more excited about is the fact that these are chiral particles. So this chiral symmetry breaking, which is combined with activity or time reversal symmetry breaking, actually results in emergence of a range of new phenomena. And I'm showing you some examples of that here. For instance, you can detect chiral edge modes in a system like this, but also more interestingly, odd material properties, for instance, odd viscosity or odd elasticity. And these are very similar to those that have been reported in quantum systems and also meta materials. Okay, so I'm going to conclude my talk. And what I hope to show is basically extracting information from the amount of data that we now can acquire from complex biological systems really requires a range of tools from multiple disciplines, and in particular physics. And I think this searching for a symmetry and symmetry breaking process in living systems can really expand this classical notion of broken symmetry in equilibrium systems to non-equilibrium systems. And I really think that it can provide us with a manageable approach to this larger question of what really characterizes the living state, or what is the order parameter of the living state. And I think this brings me to the most important slide of my talk, which is all the wonderful people in my group who have done the work. So the work that I described today is the work of a number of graduate students, undergraduate students, and physics of living systems postdoctoral fellows, in particular Zerhan Tan, who is now actually the first student graduating from my group, and he's moving on to Dresden for his postdoc. But also this has been a wonderful collaboration with Todd Gingrich and Alex Solon. I think Todd is known to the community here. And they have moved on both to their groups in Northwestern and CNRS, but also a range of different collaborations that I have with my theory colleagues, in particular Jorn Dunkel in the math department, Jordan Horowitz, who actually was a fellow, and we continue to collaborate with his group in Michigan. And finally, Erwin Fry's group at LMU in Munich, and his graduate students, Manon and Fredjof. And with that, I will be happy to take questions.
Thank you so much for that wonderful talk, Dr. Fakhri. So if you have questions, feel free to raise your hand and we will call on you or post them in the chat. It looks like there's already one question in the chat from Peter Haugen. So Peter asks, with regards to vortex pair world lines, if pair swapping is allowed, is it possible to not make a closed loop if the vortices are tracked long enough in time?
I think these are all very interesting questions. It's a very interesting question. So let me say that right now we're kind of like we're looking at the system. We have very little control in the system. So what we do is basically we just overexpress kind of like our activator, and then we kind of like we end up in this turbulent like state. And I think a lot of these questions we will be able to answer them when we actually up to genetically control like how these defects can kind of interact with each other, how they can braid or kind of form, get created or annihilated or braid. So I think what I envision is we can kind of generate a bunch of this different dynamics by tuning the different parts of the biochemical pathway. But for now, everything we see is basically what the system is giving us. So you have creation events, and then every time you have same opposite charges, they get annihilated. And I think if you have long-lived pairs, basically they just braid. So to be seen if we can really tune the system to do like some of this interesting, cool dynamics that you would hope to see, like for instance in quantum systems.
There's a question from Christine. How are persistent dipole pairs identified, and are they significantly different from the pairs that do annihilate? Are they expected to annihilate at some point?
Yeah, so basically to kind of identify persistent, so there is basically an algorithm to look for, so take one defect and look at close neighboring defects and look at basically its interactions over time. So you can kind of like identify basically these pairs looking at the nearest neighbor and how its dynamics in time. And I guess the question, the second part of the question was if, sorry.
Are they expected to annihilate at some point?
Yeah, so I think if you observe the system long enough, I guess it is possible that they get annihilated. And also they get annihilated. So they usually get annihilated when they get closer than a certain distance. I didn't show that, but you can actually show that there is a critical distance. When they get close, they basically get annihilated. So in the state where you have a lot of these defects, you do have more of them coming close than this distance. So you get more of the annihilation events. But when you have like states with sparse defects, usually they're more long-lived. But then again, it's, I mean, these are experimental systems. So depending on how much you observe the system, you basically, this is what you get from the dynamics. But yes, if you wait long enough, you can have annihilation events.
There is a question from Sherwood Richards, who is raising their hand. So go ahead and unmute yourself.
Hi, this is fantastic work and fantastic visualizations too. It's very interesting to me because I have neutrino simulations that look like they have a lot of these features, but I have not identified them in the way that you have. But my simulations are three-dimensional, so I'm wondering if there's three-dimensional extensions to the measurements of topological defects that can naturally be extended to that.
Absolutely. So what I talked about is really a two-dimensional system, but in general, topological defects are like identifying topological defects in active matter fields is a very kind of like exciting field. And a colleague in UC Santa Barbara, Zvonimir Dodgik's lab, they actually have this system where So there you do have different topological defects. So there are basically nematic defects, and these are minus 1/2 and plus 1/2 defects. They can actually generate them in 3D. And there you also have like this really beautiful dynamics, which is also a 3-dimensional kind of structure. And so yes, I mean, for this particular system, this is a 2-dimensional system, but indeed it's possible to have these defects also in 3D, again, for different active matter systems, but kind of, yeah, it's possible.
Wonderful, thank you.
There's a question in the chat from Edward Strace asking whether you can get, with these world lines that you showed, 3 of them braiding together.
Yes, that's certainly, I think these are really a kind of very exciting questions. So we're trying right now, what we are trying is, so I really think we would be able to do that when kind of like when we can have more control over the system. Right now it's basically we have to identify, like we have to look for them and then try to come up with algorithms where you can identify, okay, can you get 3 pair defect braiding. So I think the global braiding that I showed you is basically braiding on a kind of like on the whole entire surface of the cell. But then I think when we so right now my group is developing optogenetic tools where we can control like where the spirals emerge and how you can control it and how you can braid it in time. And I think that's when we can hope to really kind of do some of this exciting, basically look at some of this exciting dynamics where you can basically braid defects in 3 pairs or like multiple pairs or more complicated type operations.
But you haven't noticed any, you haven't seen any or observed any.
So so we do, but they're more anecdotal. So you really have to look very closely with it, yes. But we do have examples of that. But again, I think they're very anecdotal and kind of like it's, I would very much like to have control over being able to kind of like do it, generate them, and then kind of look at their dynamics.
Is there a theoretical limit of how many you could have? I don't know.
Yeah, so one thing actually I mentioned is basically this system can be mapped into a complex Ginsburg-Landau, I mean, it's a complex CGL equation. And we've been doing a lot of like my colleague Jorn Dunkel has been really trying to push this, push basically this CGL simulations to try to come up with different ways of like doing this type of operations. And then I think their findings will help us to really understand, okay, how we can experimentally design the system. But so what we do is we basically map it into a CGL equation.
Christina also asks that I would love to know if there's anything interesting about the time length of the world line loops.
About the time. Yeah, so okay, just first of all, I think so what, again, like what we are looking at is we so we create the system and then there's a certain time scale that you can look at it. I would say this is actually this is a beautiful system because it gives us enough time scale to, I mean, obviously these are experimental systems, so you always are limited by the system size, both in time and space. So like at least in the kind of like in how much we can push the system right now, it's I think there is a lot that we can kind of see that's interesting. Maybe I just go back to one slide. I briefly mentioned if there is anything in the time domain, and let me just point out that if you look at this X time or Y time slices, you actually see something that looks like smectics. So in the language of soft matter, like polymer physics. So I think these are like time in the time domain, and I think they're like you actually can observe defects in this time, like space-time domains, and like again, like we are trying to understand, okay, like what can we learn from this? So I think there's a lot in the time domain as well. And yeah, and the more we can push the time axis, obviously, it gives us more, I guess, freedom to explore different phenomena in the system. So, yeah.
Yeah, I was just wondering if you had like enough of a time scale to be, and also enough, I guess, of a spatial scale to be able to get a distribution of like the time lengths of the loops and see if that is anything interesting, I guess.
Yeah, so actually you brought up something interesting. So yes, we can actually look at, so you do. So if you look at, for instance, the perimeter of these loops, like in all this space-time perimeter of the loops, you can actually get really interesting scaling laws. So this is, so I'm not showing it here, but indeed you can get interesting scaling laws from like the space-time kind of loop structures. And so I think where we can push, I mean, again, like it's not a theoretical system where you can get orders of magnitude for a scaling law, but it is like within the experimental limitations, I think you can observe scaling laws even basically from these loop structures.
Thank you.
Sure.
Jamie Simon asks, well, first says, thanks for the talk, and asks, for the vortices with charge +1 and -1, is the angular speed with which waves rotate around the singularity always the same?
So yeah, so okay, let me just, so one kind of thing I have to mention is basically these are, okay, so these are, I guess, so the velocity is basically kind of like the gradient of the space field. So I don't think you can kind of like, so I mean, you can come up with like an effective type vorticity or velocity to describe it, but it's really bound to how the phase field is rotating around your defect. And kind of like one thing you see is basically the, I think that's why we kind of like say that it's very similar to, so there is really no momentum here. It's basically looking at these defects in a biochemical field or structure. So kind of there's no, so it is turbulence, but it is turbulence not in this kind of like the momentum conservation. There's no momentum here. I guess, I don't know if that answers your question, but yeah.
Maybe to clarify a little bit, when watching the videos, like in time, like watching a single vortex, the phase would sort of rotate around with some angular,
yeah,
like at some rate, right? Like
yes.
I get that it has
Yeah.
to rotate by 2 pi when you take a single time
Yeah.
slice, but also it was changing
Yeah.
at some temporal rate. So like, is that rate always the same for each vortex? Or does that make sense?
Oh no, I think that so that makes a lot of sense. So you do actually, that really depends on the state of the cell. That's actually something I'm excited about. So we don't really understand what controls it, but from state to state, it's actually it's different. So, and again, like at the moment, I don't know what controls it. It has probably something to do with the kind of like the rates that we have in this biochemical signaling pathway. But that's actually one of the things that like we're learning from the CGL kind of system to try to see if we can control it. Like in CGL, you basically can change that like easily, like you can, but here we try to understand, okay, how can you experimentally control that? But it does change from system to system.
Okay, cool. Thank you so much.
Sure.
So just keeping an eye on the time, it is 5:20. And so I would say let's all collectively thank Dr. Fakhri again for a wonderful colloquium. And if you have still some time to stick around, if anybody wants to stay on the call and ask questions, we can do that.
Thank you so much.
Well, thanks. Yep. Well, thanks for having me.
Thank you.